A very good morning, my dear students. In this video, we are going to learn about what is a DNA replication and DNA replication in prokaryotes. Okay. DNA replication in prokaryotes. What is a DNA replication? DNA replication, which is nothing but propagation or copying of DNA. Okay. The bacteria multiply by process of binary fission. Before this occurs, each cell must duplicate its genetic information so that each daughter cell has a copy by process called DNA replication. Okay. What I told you in brief, this is the explanatory form. Okay. So what is a DNA replication means just copying of genetic information from one cell to another cell. Okay. So after that only the bacteria will multiply through binary fission. Okay. DNA replication, this is the process of producing two identical copies from one origin. This biological process occurs in all living organisms and is the basis for biological inheritance. So, when the DNA is copying, the inheritance will move on to the next generation. Okay. Okay. This is the replication. So first, uh, what is that? Replication, then transcription and translation. So on the whole, which is called as this is the central dogma of life, and molecular biology, we are collectively called this term as a central dogma of life. It's nothing but replication. Replication means from DNA to DNA. It's nothing but, I just told you, it's a copying technique. So from DNA to DNA, that is replication. And again, DNA to RNA. Just transcribe the information from DNA to RNA. So this is the transcription. And RNA to protein. So just translate the information from RNA, just converted in the form of protein. So this phenomenon is called translation. Okay, so on the whole, this is the important for each and every life. So that it is called as central dogma of life. Okay. Here DNA is a template. DNA polymerase 3 replication proteins, DNTPs, ATP, magnesium ions. So for DNA replication, these are all the elements which is going to help the DNA replications. And here this is the transcription. DNA is a template, and end product is RNA. So that from we are going to transcribe the information from DNA to RNA. So here the enzyme we use is RNA polymerase, and transcription factors NTPs, magnesium ions. So these are all the factors which is going to help for transcription. And for translation, mRNA is a template, ribosomes, translation factors, amino acids, tRNAs, synthetase, ATPs, GTP, and magnesium ions. These are all the factors which is included in translation mechanisms. Okay. So these are all the basics. Okay. General features of DNA replication. So DNA replication is an extraordinarily important and complex process upon which all life depends. DNA replication is a semi-conservative replication. During DNA replication, semi-conservative means newly synthesized strand with a parental strand. Okay. During DNA replication, the two strands of double helix are separated, each then served as a template for the synthesis of complementary strand according to the base pairing rules. So during replication, what will happen means two strands of DNA double helix are separated. Each of the strands will act as a parent, that is our template for the synthesizing of a new strand. Okay, so at the time, newly synthesized complementary strands are also formed according to the base pairing rules, which we'll see in next in detail. Okay, DNA replication begins at the origin of replication. So origin of replication is the point at which only the replication begins. So next, DNA is synthesized by the enzyme. So after the point of origin, a replication point will starts. The next one is what? The DNA synthesis will begins with the help of enzymes, which is nothing but our DNA polymerases. DNA replication is a bidirectional from origin of replication. So after choosing the origin of replication point, the DNA will replicate using bidirection. So DNA replication is also extremely accurate. So equally makes error about 1 in a million. See how much accurate is this. See, despite its complexity and accuracy, replication is a very rapid. In bacteria, replication rates approach 752,000 base pair per second. See how fast. And replication steps. The replication of chromosomal DNA begins at single point, the origin of replication. So you know already, at that point only the replication fork will form and continuously the replication will start. DNA replication begins with the unwinding of two antiparallel complementary strands, resulting in the formation of two single strands. This unwinding process of two replication forks, the synthesis of DNA begin at the replication forks. So once the replication fork is formed, the synthesis of newly formed DNA, it will start to synthesis. Okay, the replication proceeds in 5 prime to 3 prime directions and is semi discontinuous away from movement replication fork in short segment called series of fragments called Ogasaki fragments. This strand called lagging strand. See, the replication will start 5 prime to 3 prime directions and it is semi discontinuous away from movement of replication fork. So from the replication fork, it will synthesize like short fragments of newly synthesized fragments, which is called as our Okazaki fragments. So this strand is called as a lagging strand, which means a lagging in between some lagging portions are there, so that it is called as a lagging strand. Of the two strands, one strand is synthesized continuously in the direction movement of the replication fork. So the previous one we are saying that the lagging strand, which is in between some lagging portion is there, some small newly synthesized fragments are here. So that small newly synthesized fragments of DNA is called as Ogasaki fragments. That strand is called as a lagging strand. And again, here the continuously synthesized strand from 5 prime to 3 prime direction is called as a leading strand. While other strand is synthesized, it is continuous. Okay. Two replication fork proceed in opposite direction from the origin until they meet at a site called replication termination site. Once the replication fork is started at the two sides, it will meet again at the again until the replication termination process. Okay, so at that site is called as a replication termination site. See, the first diagram A, that is 5 prime to 3 prime, one arrow mark Okay, and again 5 prime to 3 prime opposite direction. So here two replications are start in bidirectional. Okay, first diagram describes it, and the second one shows, see, I'll use the pointer. See, this is a 5 prime to 3 prime direction, and here also 5 prime to 3 prime direction. One is this way and another one is this way. So this is a bidirectional replication. So here is the origin of replication. So once the replication fork is find, and the replication will start this way and this way. So this is a bidirectional replication, and see the B diagram. So it will extend. So initiation and elongation. It will extend. Okay. See the extension. And see, this is the, what is that? Some short fragments in the size, and here also some lagging, and here is some continuous. See, here is the, there is no lagging. This is a continuous synthesis. So this is the leading strand and this is the lagging strand. Okay. See, this is the, so for E. coli, the circular DNA is the origin of replication starts here. Once the origin of replication starts, the replication fork, it will unwind the double helix. Then see, as I told you already, once the replication forks will unwind the double-stranded DNA, and further it will meet at the termination site. Okay, site where the replication ends. At the time only, these two replication fork will meet. Okay, now hope you clear. DNA replication involves the action of number of specialized enzymes and proteins. See, this is one important part of this DNA replication. This is the enzymology of replications. What are all the enzymes which is involved in the replications? What are all the proteins which is mainly used for this replication process? See, first one is a helicase, which is unwinds the double-stranded DNA, which is separates the double-stranded DNA. DNA topoisomerases, which relieves the topological stress produced in the helical structure of DNA during unwinding. See, if you take one rubber band, if you twist it bidirectionally, and you have to mm, pull the center portion, what will happen actually? Some stress you may feel at double ends. Okay, so that is the topological stress. So this topoisomerase, which relieves the topological stress produced in the helical structure. So the helical structure, as we all know, when we are twisting again and again, some stress will occur. So this topoisomerase, which is the enzyme which relieves the stress during unwinding. Okay, when separate the double strands, definitely some stress will arise. So it will release that. Okay, first one is the DNA polymerase 1, and this is the DNA polymerase 3, and this is the DNA primase. The name itself, it can the DNA primase means synthesis of primers, is an enzyme which is involved in replication of DNA and is a type of RNA polymerase segment called primer complementary to a single stranded DNA template. So primase means is a type of RNA polymerase. The segment called a primer complement is a synthesizing primer. The primer which is complementary to the single stranded DNA template. DNA ligase. Ligase means nothing but can help to ligate. Catalyze the formation of phosphodiester linkage, which catalyze the formation of phosphodiester linkage. Through that it will ligate the molecules. and single stranded DNA binding protein, that means SSB. So stabilizes single strands of DNA after unwinding. So after unwinding, after separation of double strands, it will help to stabilize the single strand. Okay. So these are all the enzymes and proteins which is mainly involved in the action of replication process. Okay. See this diagram. So previous slide I show you the ABC diagram, like Y structure of ABC diagrams. So here it is in detail. So see this leading strand, and this is the lagging strand, which is called Ogasaki fragments. That is short form of newly synthesized fragments. Okay. See, this is the DNA polymerase, and this is the parent DNA. See, this is a single stranded binding proteins which gives the stability for the single stranded DNA. Okay. Primase, and this is the DNA polymerase, and another DNA polymerase is a ligase which helps to catalyze the phosphodiester bond, through that which ligate the molecules. The DNA polymerase. DNA polymerase is an enzyme that carries out the synthesis of new strands or the template strand. From the template strand, it can be used to synthesize the new strands, a daughter strand. The enzymes called DNA polymerase catalyze DNA synthesis. Catalyze DNA synthesis. All known DNA polymerases. DNA polymerases catalyze synthesis of DNA into 5' to 3' directions. The nucleotide to be added is a deoxynucleoside triphosphate, DNTPs. Okay. From these DNTPs, the nucleotides are added to the complementary strands. Deoxynucleotides are linked by the phosphodiester bonds formed by the reaction between the hydroxyl group at 3' end of the growing DNA strand and the phosphodiester closer to the 5' carbon of the incoming deoxyribose. DNA polymerase reaction. The hydroxyl of the 3' terminal deoxyribose makes a nucleophilic attack on the A phosphate to the nucleotide substrate. See, 3' terminal makes the nucleophilic attack, which is nothing but our replication is the 5' to 3' directions. So what will happen automatically, the nucleotides or DNTPs will add on to the 3' terminal. Okay. So these are all the DNA polymerases in E. coli. There are at least 5 DNA polymerases associated with E. coli DNA replication. These are DNA polymerase 1, 2, 3, 4 and 5. So first one is the isolated DNA polymerases to be isolated and purified. The polymerase has 5' to 3' exonuclease activity. Exonuclease means outside the nucleus. Okay. So outside the nucleus, it has the 5' to 3' exonuclease activity. So DNA polymerase 2, the enzyme is encoded by Pol B gene. The polymerase 2 enzyme is encoded by the Pol B gene. It has no 5' to 3' exonuclease activity. Rather, it has 3' to 5' exonuclease activity, which is a proofreading activity. See, as we all know, the replication starts from 5' to 3' directions, but the DNA polymerase don't have that exonuclease activity at 5' to 3'. Instead, it has 3' to 5', that is reverse. So what the enzyme will do, which is automatically do the proofreading activity. Okay, so it can easily detect whether it is correctly paired or not. Okay, so DNA polymerase 3, this is the principal replication enzyme, which is This is a principal replication, not a basic, the principal replication enzyme encoded by the gene POLC. It has 5' to 3' proofreading activities. See, it has polymerase 3 has 5' to 3' proofreading activities. It lacks the reverse, that is 3' to 5' exonuclease activity. See, DNA polymerase 2 and 3 has a alternative mechanisms. It has very high processivity. DNA polymerase 4 and 5 are involved in an unusual form of DNA repair. So for DNA repair mechanism, these two enzymes are involved. DNA polymerase 3. DNA polymerase 3 holoenzyme is a primary enzyme complex involved in prokaryotic DNA replication. DNA polymerase 3 synthesizes base pairs at a rate of around 1000 nucleotides per second. See, wow. As the replication progresses and the replisome moves forward. Once the replication progresses, the replisome moves forward, DNA polymerase 3 arrives at RNA primer and begins replicating the DNA, adding on to 3' OH of the primer. Okay, so as I told you already, 3' end has the nucleophilic activity, so the nucleotides will add on to this site only. So DNA polymerase 3 will then synthesize a continuous discontinuous strands of DNA. So continuous strands, as we all know, the leading strand, the discontinuous strand will know that is a lagging strand. So DNA polymerase 3 has a high processivity and therefore synthesizes DNA very quickly. This high processivity is due in part to the beta clamps that hold on to the DNA strand. DNA replication in prokaryote. So DNA replication in prokaryote, which comprises of these many steps. Yes, we all know the E. coli has replicated bidirectionally from a single origin, that is origin of replication site. The circular with no free ends replication of DNA in E. coli is also known as theta replication. So what is the replication in E. coli? Theta replication. Why it is called as a 3 theta replication? I just shall I show the diagram? Okay. See the symbol which denotes like theta. So E. coli chromosomal DNA just look like a circular DNA. So once the origin of replication find, it will unwind the double-stranded DNA and the parental DNA synthesized with the help of template DNA. Then only we'll call it as a theta replication. Okay. Now we can move on to the next one. Okay. So first one is the initiation, second one is elongation, the final one is termination. So see, these are all the proteins and enzymes which is involved for the DNA replications. The sequence of events during initiation. The E. coli ORC consists of two short sequences. One is where DNA A protein binds and other is called DNA unwinding element. DNA unwinding element. The binding of DNA A, this is the protein which facilitates the initial strand separation of E. coli DNA duplex. Okay. With DNA A is the protein which facilitates the strand separation. DNA C protein binds to the DNA B protein which results in the opening up of DNA B ring. The two DNA B ring, one of each strand. DNA C is released and two DNA B remains bound to the two single-stranded DNA. DNA B helicase is then loaded on two single-stranded strands which then travels along the single-stranded DNA in the 5' to 3' direction, unwinding the DNA strand, creating two replication force. Unwinding produces stress, which is removed by DNA gyrase. Unwinding produces stress, obviously, yes. It is removed by the enzyme DNA gyrase. So finally, the origin of replication, that is RC site DNA, is methylated by DAM methylase. See, the entire description will shown in one figure. See, unwinding origin. RC DNA is a protein which helps to unwinding the primosome. Primosome, it has a DNA C and DNA B protein which helps to assemble the single strand. Okay, assemble the single strand. After unwinding, these proteins helps to assemble, and polymerase 3, see the helicase, the protein which has DNA B. And this is the DNA polymerase 3. So once the replication fork starts, the single strand was hold with the stability of these proteins, then the replication will start from 5' to 3' directions. Okay. Elongation. In this phase, the synthesis of two new daughter strand takes place complementary to the template strand. DNA polymerase 3 is the enzyme that synthesizes the daughter strands. At this point, a primer is needed so that the DNA polymerase 3 can begin to act. A primer is a short segment of RNA that provides 3' OH group to which the nucleotide can be added. This phase is marked by synthesis of a leading strand and lagging strand. Leading strand is synthesized continuously in 5' to 3' direction, and in contrast, lagging strand are 5' to 3' direction, but it's not a continuous synthesis. Discontinuous synthesis by loop formation in short segments called Okazaki fragments. See here is the elongation process. This is the Okazaki fragments, this is the lagging strand, this is the continuously synthesized leading strand. Okay. DNA B helicase first unwinds the replication fork. See. Here DNA B first unwinds the replication fork. The DNA primase then associate with the DNA B helicase which synthesize short RNA primer. The primer is then extended by the DNA polymerase 3 which completes the synthesis of one Okazaki fragments. Here the DNA primase is associated with the DNA B, then form the, with the help of polymerase, DNA polymerase 3, it will form the short Okazaki fragments. Once an Okazaki fragments are being completed, its RNA primer is removed and replaced with the DNA by DNA polymerase 1. And the remaining nick is sealed by DNA ligase. See. See, once the Ogasaki fragments are synthesized, the RNA primer is removed, which is replaced with DNA polymerase 1, and the remaining gaps were filled with the ligase. Okay. DNA ligase catalyze the formation of phosphodiester bond. Okay. Next, the final step is termination. Replication of bacterial genome proceeds bidirectionally, which terminates at a position diametrically opposite directions. Replication terminates at the terminus region containing multiple copies of 20 base pair sequence called ter sequence. Okay, replication terminates at the termination recognition terminus region containing multiple copies of 20 base pair sequence. It is a ter sequence. The ter sequence works as the binding site of protein, test protein, that is terminus utilization substance, test protein, which stops the DNA B helicase, resulting in the formation of DNA replication, resulting in the termination of DNA replication. See, the test protein which stops the DNA helicase synthesis and resulting in termination of DNA replication. The completed chromosomes then partition into two daughter cells during cell divisions. So once the replication stops, the complete chromosomes then partitioned into two daughter cells during cell divisions. So see here the ter sequence, there is a 20 base pair ter sequence. See, we just start here and end here. Okay. It's a TUS protein which terminate the DNA helicase synthesis. Through that will terminate the replication. Okay. So this is the overall process of DNA replication. Initiation, elongation, termination. So initiation with the help of enzymes, proteins. These are all the one of the important thing which is present in this process. The elongation and the termination. Okay. So these are all the process which is present in prokaryotic DNA. Okay. Thanks for your attention. Thank you.